Hey guys. Can someone please explain to me what's happening outside our windows right now? It's so terrible. I just bought all these plants at Cook and Gardner and got them in the ground. Okay. So I don't know. I wanted to do that parent share, but we're low on people right now. So I don't know how that works when people join after you've set up breakout rooms. Maybe you just assign them separately. Anybody have a clue about that? Anybody had that experience in class? No? Okay. Well, let me kind of go over what I was thinking about doing, and then we can stall for a little bit for others to join. All right, so if you guys can check out your chat, I'm going to share a link. One sec. Okay, so click on that link and let me know if you see a Google Sheet. Are you guys seeing the same thing I have up? Okay. All right. So I think what I'm going to have us do is I've got two slides. Again, it's in the chat, but it's also on our Moodle site. So I've got two slides. One generally talking about the rates that we were talking about with island biogeography, and then a visual here of an island archipelago and some trends. And then our next slide kind of gives some, variations on what those might be. I don't want to spend any time. I want you guys to talk to each other about it. So what I'm going to have you guys do is, and again, some of you I think jumped on a little later. Let me copy this link out. I'm going to pair you up. I want you to spend 5 minutes explaining the concept and the trends on those slides together, and then come back, and then I'm going to show you kind of where the potential to get the extra points and stuff will be. It's not available till after class though, I think. All right, so let me get you into breakout rooms. And again, just 5 minutes. So. Okay, see you in a little bit. Andrew, are you there? This is to earn some extra points, Andrew. Hopefully you're here. If so, join the group. Chase.
Oh, hello.
Hey, I'm going to send you to a breakout room, okay? Just briefly.
Okay. Okay.
Graylin and Jordan, are you guys good? Thoroughly understanding?
Yeah.
Yeah, good, good.
All good.
Okay, well, stay tuned for everybody. I just hooked somebody up to a group, so give us a sec. Devin, you were by yourself for a little while, right? I realized you were all by yourself.
My Wi-Fi is really bad right now, so I got like booted from my first breakout room and then tried to come back and then was just alone.
Oh no. Did Chase join you shortly thereafter?
Yeah,
Okay.
and then Chase found me and then we
Okay.
had a moment
Yeah.
before the break.
Okay. All right. So everybody's back? I think everybody's back. Good. Good stuff. So let me show you guys what I'm hoping will be a good thing for you. Okay, so later today, all the way through tomorrow, if you guys can go to our page and it's like the slow as molasses here. Go down to where the quizzes are. So there's going to be a redemption points, island biogeography. Okay. So it should be, if all goes okay, an opportunity for you to take like a little mini quiz, I guess, and earn some points back on the test. I'll just be adding them to like our numerator of the overall grades. So not necessary that you do it. If you want an opportunity to reinforce what you just taught each other and what we've talked about a couple times now, again, coming from the landscape ecology unit, maybe trickier material, then feel free to go in and do that. But you'll have, I think I let 20 minutes once you've started. It's only like 4 or 5 questions. So they're all regarding what you guys just covered. So anyway, it is there. It's down in quizzes, redemption points. And hopefully you guys take advantage of that. Okay. Couple things. Firstly, so yesterday there was a really big verdict, right? I don't know about you guys. I wanted to take a minute and acknowledge it. I was really emotional about all of it. So not that I am the be all end all expert on moving forward and justice and peace for this family and everybody who's sort of moving in towards equality and fairness. And again, I just want you to know that I am here. I am feeling it all, just like you guys are, and I am available to listen and talk with you if you want. Again, just a really emotional time. I talked to my kids about it too, and I just wanted to let you guys know that I'm there for you if you want to talk. So it was a big day yesterday. Okay. Also, this is nonsense outside. Tomorrow is Earth Day, right? And it's snowing. It's kind of a slap in the face to all of us. So we have a poster session tomorrow afternoon. It's falling directly on my office hours, which run 4 to 5. So if anybody needs me earlier, I'm done teaching, by 1. So sometime between 1 and 3:30, you guys could reach out via email and I can coordinate a Zoom for you if you need me tomorrow. I'm just going to probably not be able to be in the office hours tomorrow afternoon. Okay. Good. I feel like a bit of catharsis right now. So anyways, all right. So I tried so hard, you guys. I wanted us to have a moment where we could watch a darn video. And it is an amazing video, and I'm going to still do my best to make it happen. My CD-ROM, which is like 1985 computer, is pixelating it and giving us like the worst volume is like every 15 seconds you hear a word. So I'm going to try and have the spouse rip this tonight and get it onto a server somehow so that we can watch it like, and an enjoyable experience as opposed to that. So stay tuned. We're just not going to be able to watch the video today. I don't know why the CD-ROM is giving us trouble, but anyway. All right, so in place of that, we're going to just kind of keep trucking on our ecosystem lecture. And like I said, I'll pepper that in as soon as I possibly can. So everyone's seeing nutrient cycling? All right. So nutrient cycling is Dr. Aldred's jam. It is not at all the level of ecology that I focus on, but I can tell you that with my research, I've really had to do a lot more soil work than I ever have done in the past. So, you know, I'm a wildlife ecologist by training, but because I'm part of this Ecological Researchers Education Network, I've started working on a lot of plant-based projects. So I do a milkweed survey. I've done an invasive garlic mustard survey, and I've done some pea and corn experiments and so on, looking at allelochemicals. And anyways, having done that, I know that it's really important for us to look at soil moisture and soil pH. It's important for us to evaluate levels of nitrogen and phosphorus compounds in the soil, because it all enhances or reduces the potential for the plants to perform well and photosynthesize and grow and set seed. So we're going to be focused in this unit, in this particular lecture, on watching the pool of those, you know, C's, carbon and nitrogen, phosphorus, where it moves through the system, how it goes from the soil or from the atmosphere into the organism, and then how it moves through that food chain and returns itself either to the soil or to the atmosphere. So that's the focus we're going to have with nutrient cycling. So we'll talk about inputs and outputs. We'll talk specifically about a handful of the cycles, which should be kind of basic review, I think, in most cases, but putting it in context. So again, ecosystems, we're watching how the abiotic and the biotic interact and feed into each other. And we're going to be looking at the origins of those cycles. So where do those nutrients derive from? Are they coming from the atmosphere, typically the gaseous phase? That can be from the ocean derivatives or the atmosphere, but mostly atmospheric, like, for example, atmospheric nitrogen, the N2 gas, which is the majority of the nitrogen that we have, is unusable except for certain organisms like bacteria. And then sedimentary, which derives from erosion, rocks, and erosion and guano and so on and so forth. But these are really, if you want to know where those nutrients are coming from, these are the two pools, either gaseous or sedimentary. And when we think about those nutrients moving through our system, they really are defined and moving through two different processes. These are the two processes that are really, really important, and we're going to be focused in this lecture on, you know, what makes those processes go faster or go slower. So the first one is productivity. Think about making things, right? So we're talking about, is Devon's chilly? She's getting her jacket. So productivity is, you know, taking the energy, transferring that inorganic material, which will typically, you know, be drawn up through the soil in many cases and transformed into organic material, usually into plants. And then decomposition is the opposite end of the spectrum. So this is going to be the process where those organic materials will be decomposing and broken down with the help of fungus and bacteria and so on, and bugs, right? Turning it back into the inorganic nutrients that will go back into the soil and start the whole process again. So two ends of the spectrum, two processes. These rates are production or productivity and decomposition. And the speed at which they move is a function of temperature and moisture, which we've covered before, as well as the quality of the material. So the quality of the material, think about, remember we talked about like beech leaves, when we talked about bottom up, beech leaves had a lot of lignin. They're really tough and they don't break down very easily. So you could be walking in the woods 10 years from now, maybe even see beech leaves, as opposed to maple leaves, which get slimy and bacteria biofilms grow on them really, really readily. So they'll break down a lot faster. So the quality of the leaf litter, as well as the abiotic factors associated with the conditions.
Danielle, I have a quick question.
Sure.
How, you know, bay leaves, which you use to cook?
Yes.
Why do
have
they
a lot
not
of
break
lignin.
down? Is
Yes,
that what's in them?
Yep.
Okay.
Yep. They have a lot of
They're
that
still
structural material.
solid after you cook with them.
Yes. Yes. And they are an excellent addition to any soup that you make. So I highly recommend
Yes.
bay leaves. Yes. Or lentils
Cool.
too. They're really good at lentils.
Mm.
So good question. So decomposition certainly does require the help, or can be aided with these little microorganisms. So we're talking about the nematodes, our little roundworms, tardigrades, right? Love the tardigrades, those little water bears that can be taken up into space and still make it. And down to our earthworms, which, you know, we're going to be surveying in a week or so in lab, and all those insects that I don't really care to study on my own, but other people do. But all of these things are going to be helpful in breaking them down. In addition to those invertebrates, we also have soil fungus, which can aid as well. And we've talked about it before. We've talked about it pairing up with plants in many cases to increase the rhizosphere, right? It can help pick up trace nutrients in water a little bit more. These ectomycorrhizal fungi are going to pair up with the root hairs, right? And increase that space to draw that stuff up. Now, on the test, you guys remember, this was maybe a tricky one for some. There was a question about mutualisms. Do we find more mutualisms in stressed microhabitats or habitats, or not stressed? I can't, it's the opposite, something like that. So you would want a helper when you're struggling, right? And just kind of think of it that way. So it's usually the stressed environments, the desert, the tundra, where you tend to see more instances of those mutualistic relationships, right? In other places, the relationship, if you had a mycorrhizal fungi, might become parasitic if you have all that you need, right? If your environment is okay. So again, the fungus we've often talked about in this symbiotic relationship, as I mentioned. They will certainly act just like bacteria and those microorganisms in helping to decompose, and they seem to tolerate more acidic soils than, say, bacteria can. So they can be helpful if you're in like a peat habitat, you know, like peat moss, where the acid might be a little bit more. And then finally, bacteria. So we've talked soil microorganisms, we've talked about fungus, now we're focused on bacteria. Again, we've talked about bacteria in a symbiotic relationship, a mutualistic relationship. Those rhizobia, right? We've got soil bacteria that get uptaken by the root hairs, and they form these root nodules. The root hairs will form these root nodules, which inside will have the bacteria, right? Those are called rhizobia. And to be honest, I just dug up a whole bunch of mulch as I was planting the flowers that are now dead because of the snow. And there were rhizobia attached to old root masses. You know, we were chipping a whole bunch of stuff, and it's been sitting out there for, 3 years now. And I'm like, I'm just going to use it. The carpenter ants have killed all of our trees. So when the trees fall, we just chip it. And now I'm gonna thank you, carpenter ants. I don't have to pay for mulch. But this stuff was all in there. And again, the nitrogen that a plant needs as a limiting nutrient in order to photosynthesize, in order to max up that NPP, right, net primary production, the plants can't like fix it into a form they can use, right? So the bacteria can. The bacteria can take that N2 gas and convert it into something like nitrate, nitrite, or ammonia. And then the plant can take it up in their roots and do, maximum photosynthesis as a result of it. So these bacteria are really, really important. They're the ones that are kind of the ones that can convert more so than maybe any other organism. And when we get to the nitrogen cycle, you'll totally see that. the majority of the nitrogen cycle is all taking place underground with these bacteria. Okay, so other things besides, you know, having those organisms around to help in decomposition that will influence the rates here are, as I mentioned, temperature, water, and the availability of oxygen is kind of a new one. So think about like being totally submerged in the water and having no available oxygen. Some bacteria, you know, are aerobic and need that. And so if you have oxygen, then maybe there'll be faster rates of decomp. But litter quality is the one we haven't really focused on, right? If it's warmer, if it's wetter, usually we have, you know, higher rates of decomp. But we haven't talked about the litter quality yet. And so we'll get to that in a sec. So organic matter accumulation. Let's walk through a couple of the biomes that we've covered. So when things decompose, they can either be immediately uptaken into the plants again, right? That's what we saw with the tropical rainforest, right? As soon as things break down, they immediately get sucked back up, which is why, you know, clear cutting in the tropics is bad. But in the grasslands, there tends to be pretty low moisture. Seasonally, it can be a little cold, which inhibits the fungus and the bacteria from breaking stuff down. So what we find is that if you were to like dig a pit in a prairie system, a grassland prairie, you will find a lot of soil organic matter and a lot of, you know, piles of roots, you know, year after year, just kind of slow to decompose. We'll also find a similar thing in peatlands. So this, you know, think maybe boreal forest where we find these peatlands. What we find here is the rates of decomp, again, colder temperatures, a little bit more acidic soils because it's peat, right? And less oxygen in the soils for the soil microbes to use and thrive on. So stuff tends to accumulate permafrost, right? Think about our permafrost over time, the peat building up into that and then freezing because it's colder. And so they found bodies that have been preserved in these peat bogs and stuff. It's pretty crazy. If anybody's ever like on a trip, that's pretty disturbing. If anybody's ever on a trip to Pennsylvania and you go to Philly and you're doing, you know, the typical like tour, I'm gonna go see the Liberty Bell and stuff. I encourage you, and maybe it's because I'm weird, to take a couple of hours and go to the Mutter Museum. It is a museum of interesting biological, let me see if I can pull it up here. But I think they have these peat people. The Physicians College. And they have all of these interesting conjoined twins and preserved, here we go, right? The preserved bog people. It just absolutely kind of fascinating medical mysteries and stuff. It's the Mutter Museum. And it's the Physicians College that is maintaining it in Philadelphia. Again, totally off topic. Well, kind of. But I think you'll see that bog lady. Okay. All right. So the amount of organic material that accumulates is going to be different in different biomes, right? So we talked about it being high in the wetlands. It's acidic, it's cold in some cases. Well, not necessarily in this case, but there's maybe less oxygen, so it builds up. Over here, it's cold, so the bacteria aren't working as much to break that down. So it accumulates over time. So in addition, there's also the idea of the leaf litter quality, right? And, you know, bay leaves, again, good example, beech leaves, but typically the non-woody plants. So you guys remember we were out, I don't know if Dr. Aldred pointed it out, but the trillium were just about to pop in the lab when you guys were out doing the trees. They're the 3 leaves, you know, trillium tri, right? So there are three leaves, 3 sepals here, beautiful flowers. They can be red, they can be white. I think there's some pink ones as well, but they were just about to pop. So any herbaceous plant, any flowering plant, grasses are quicker to decompose than the woody stuff, right? there's less, lignin. In addition, phytoplankton and zooplankton and other of those smaller bodied organisms, the ones we maybe classify as R selected, are going to be more readily decomposing than the K selected species. So daily rate of mass loss or rates of decomposition as a function of leaf solubility. The more soluble your leaves, right? If you think about if you crinkled it all up and it's still there in its entirety, that's going to probably have a low rate of decomposition, and it'll be higher rate if you can smush it all up and it'll decompose. Now, there's a lot of people that do research. It's something we might build into. I know we used to do this in general ecology lab. We had you guys sew like window screening into little baggies and then go out and stuff it with conifers and some mixed forest, like deciduous tree stuff and some with higher lignin, some with lower lignin, and then invasive species. And we would have you sew them up, and we would place them on the side of like the Chip Cummings field. We would like put a flag in it to make them stick there. And then we would put some also in the stream as we pass the gate, and look at how water changes the decomposition rates as opposed to being in the dry area. And then we would have different size mesh. So some would be more open, so earthworms could actually get in, and others would be, no invertebrates will get in, but typically just the bacteria could get in. And we'll see kind of how that influences the rates of decomp. But in general, the more lignin, the tougher your leaves, the less decomposition you're going to have. Okay. And you can see beech, we've talked about beech and oak. Those have high lignin. Elm and ash have lower lignin. But notice if you add invertebrates into the mix, you get higher rates of decomposition, right? Without the invertebrates, we're looking at leaf mass remaining. So that's like the opposite of decomposition, right? So anytime you have invertebrates, we typically have more of it being decomposed. And anytime you have a leaf that has tougher skin, tougher structure, the beech, the oak, we have less rates of decomposition as well. Okay. And again, just reinforcing it, decomposition rate is low in sycamore and oak. It's much more than that in remember we saw cottonwoods and aspen trees when we were out doing our tree surveys. Those have way higher rates of decomposition. And this here is in the tropics. We're looking at how long it takes days of decomposition, the amount of leaf mass remaining. So, the amount of leaf mass remaining, the steeper that slope is, probably the better quality it is for bacteria and fungus to work on it. This slope being less steep suggests that more of it is remaining. So I would suggest here that the trumpet tree is probably tougher leaves than, say, the Brazilian fire tree. Okay. Now think about what that means. Remember, we've been talking about bottom up and top down. The more types of trees and plants that you have that don't have as much lignin, the more available nutrients you would expect to support bottom up, right? So the quality of your forest, overstory and understory, as they start to break down, really does matter, because that's the base of your pyramid, right? That's the base of the energy that can be built up upon it. So temperature, right? If we look at percent mass remaining over time, and we look at New Hampshire versus West Virginia versus Virginia, so we're looking kind of latitudinal gradient. There is more mass remaining in New Hampshire up in the cold, right? And less mass remaining, so more decomposition in Virginia and West Virginia. Now, the rate, the Virginias are kind of similar in latitude, right? But one is more inland and one is more coastal. So the amount of moisture could be influenced as a result of that, right? And West Virginia tends to have some mountains too, so that temperature could play in too. So look at the mass remaining in leaves and roots, right? Versus the biome. And I just kind of wrote some take homes here, right? The blue, I'm sorry that I can't even see colors today. The purple is tundra and the green is the boreal. So these are the colder biomes, right? Percent of mass remaining is higher, right? So lesser decomposition as compared to a tropical rainforest. The amount of mass remaining is really low because it's a high rate of decomposition, right? And you can see that happening also for the roots too, right? When anywhere where it's hotter, you tend to have higher rates of decomposition, less mass remaining. Okay, now look at water, percent of the mass remaining over time, slower to decompose on the dry land, faster to decompose in the water, okay? And you probably, you know, have you guys stepped in a pond that's got kind of muddy, mucky bottom to it, right? You can imagine that there's a lot more microbes down there and fungus and stuff that could break that stuff down. And the conditions would be ripe for that. Now, we also talked about oxygen, right? Especially in water where oxygen can be limited. Remember, we talked about those wetland plants that had parenchyma, right? The holes that allowed for oxygen to diffuse quicker, that like unique strategy, just like we talked about pneumatophores, right? The cells that are going to allow the mangroves to take in oxygen. But these wetland plants have these parenchyma pockets, which allows for greater oxygen efficiency. So for example, here, percent mass remaining over time, if we have aquatic plants, you know, say the marsh plants, the decomposition rate is going to be much slower if we're in an anaerobic or without oxygen scenario, and we'll have a lot faster decomp, a lot less remaining if we do have the available oxygen. Okay, so that's just kind of something to consider. Anaerobic respiration, less efficient if you can have oxygen available, again with the help of things like parenchyma. Then we will have more organic matter being decomposed and the nutrients returning to those other plants that need it. Now, I want you to look at this visual. There's a lot of words here, but let's look at the picture, okay? On land versus in the water, okay? I want you to think about where productivity happens and where decomposition happens. So on land, right, production occurs, NPP created, usually in leaves, right? So you can kind of think in the canopy. So you've got spatial separation of where your organic material is being created, right? In the leaves being captured, and you're having you know, photosynthesis occurring. And then these will die and decompose and return to the soil. There's a spatial separation between where production and decomposition occurs, right? The soil is where decomposition will occur, right? Where the leaf litter kind of it falls down, the trees fall down, and that's where the bacteria and so on will work on them. And the photosynthesis occurs up in the top, okay? There's a spatial separation, however, they are connected by a root system and they're connected by a trunk, right? So the pool is all one kind of unit, right? You can kind of see how that's all one kind of unit. If we're in the aquatic system, the sediments and where the plants are, where they're photosynthesizing, are totally spatially separated, okay? There's no connection, right? The roots and the tree above where all the photosynthesis is occurring are not connected. You have floating phytoplankton and algae up here, only in the photic zone. You hardly have anything going on in the middle. Down at the bottom in the sediments, you have decomposition occurring, right? There's that spatial separation. The only chance you have of the nutrients that are needed for the phytoplankton and the algae to grow and to get the nutrients, so they're no longer limited and they can, you know, grow and have high NPP. The only time that will occur is if there's upwelling, right, in the ocean where the nutrients, the wave action and stuff, big storms will return nutrients from the bottom sediments and bring them up to the top where the plants can access them. Or, I've lost my train of thought. Or in a lake where turnover occurs, right? Remember the thermocline breaks down in the wintertime and we start to see those kind of water shift, bringing those nutrients back up to the top so those plants can access it, right? So in an aquatic system, your stages of production and your stages of decomposition are totally separated in space. On land, although there's like vertical structuring separating them, the plant itself connects it, right? The plant is rooted in the ground where decomposition occurs, and the top of the plant is where the photosynthesis occurs, where the production occurs, right? So there is that connection. So, you know, how do those nutrients cycle? They certainly do cycle differently on land versus in the water. They all will, you know, uptake the nutrients, do their business, grow, reproduce, set seed if they can. But just kind of think about the linkages between those two rates, those things that make nutrient cycling happen, production and decomposition. They are disjunct, separated in a lake aquatic system, you know, but they are connected more so in a terrestrial system. Oops. Okay, so this idea of Liebig's law of the minimum, you might have heard it before, but as I've mentioned, right, if we don't have water, your plant's going to wilt, you won't photosynthesize well, right? If you don't have nitrogen in some cases, your plant's not going to be able to photosynthesize as well. And remember, we talked about 3 different places, right? Terrestrial systems, freshwater systems, and the marine systems. We mentioned there were certain nutrients that those places lacked, that if they didn't have enough of it, the plant wouldn't photosynthesize and they couldn't crank out the NPP, right? So we mentioned in the ocean, we need nitrogen, we need silica for the shells of diatoms and stuff, and we need iron, right? The iron filings, we talked about that with geoengineering. If we've got all those nutrients, we can crank out our photosynthesis and production. In freshwater systems, organisms tend to be phosphorus limited unless there's too much of it and we're eutrophied, and then nitrogen will often become limited, right? And on land, it seemed to be that we needed both nitrogen and phosphorus to really crank up that production, right? So it's really important to note that, you know, certain biomes, certain habitats are going to have different nutrients that will limit their ability to produce, okay? And why do we need certain nutrients? So you need nitrogen because nitrogen's the backbone for a lot of the, you know, nucleic acids in your proteins. You need carbon because that's part of the structural material of a plant and other organisms. You need phosphorus to help in running the chloroplasts and those powerhouses, the mitochondria. Okay, so, you know, think about an organism, think about all the cells and vacuoles and so on, and there is a reason why you need each of those nutrients, okay? And nutrient limitation is, again, what Dr. Aldred does evaluate. And we talked a little bit about this with the bottom-up discussion, right? If we add fertilizer so that we're not limiting nutrients anymore, what we found was we ramped up biomass, we ramped up the abundance, the number of individuals, but we reduced the species richness, right? Over time, keep adding nutrients, we ended up with just, you know, a monoculture of one particular species that tolerated a lot of nutrients. It was at the very beginning where we had the most species, but anytime you add fertilizer, add those nutrients, you tend to maximize the number of individuals that will grow, especially the ones that could tolerate that nutrient profile, right? So Dr. Aldred looks at how, you know, net primary production or your productivity varies as a function of nitrogen input. You know, if you don't have a lot of nitrogen, you're not going to have a lot of production. Crank it up and you will, but to a point, right? She also looks at how, you know, photosynthetic rate can change as a function of the level of nitrogen, and again, measures heights of plants and so on. So where do these nutrients derive from? As we mentioned, we talked about there being an origin, either atmospheric or sedimentary. Well, phosphorus is one of those sedimentary ones, okay? If you had to place where it comes from, it's going to be from the rocks, okay? And so we've got mining, we've got wastes, so the guano that a lot of the, you know, birds that are out on rocks will be rich in phosphorus. We've got the the aquatic organisms, as they die and decompose, they return phosphorus back into the soil. And so the majority of what we would consider is that it's going to be locked up in sediments, and that over time, due to weathering, phosphorus will be accessible. Okay. And that's usually the one that's limited in some of those lake systems that we talked about. So again, where is it coming from? It's coming from erosion of the rocks, mining of the rocks, guano, as I mentioned, you know, the waste of these organisms, and winds can carry it across on the jet stream to other places that you wouldn't think have it. And so we tend to see that in really old soils that are highly weathered, they have lost a lot of the phosphorus. Okay, the newer soils have more phosphorus. So for example, if we looked at the archipelago of Hawaiian Islands, there are some of those islands that came first, and some that are, you know, older, and some that are newer. So the young soils in Hawaii have higher phosphorus. And the older soils, like Kauai, which is an older island derived, you know, rose up earlier, has lower phosphorus. Okay. Now, nitrogen. I mentioned nitrogen. There's a lot of important bacteria involved in nitrogen. I don't need you to memorize these structures. It kind of freaks me out a little bit. It's very organic chemistry right here. But these are all proteins, right? As I mentioned, nitrogen really important in proteins and nucleic acids. And we've talked about a really important protein or enzyme, right? And that was rubisco, right? In photosynthesis, and that our plant leaf is really action-packed with rubisco, right? 50% of the nitrogen in the leaf derived from this particular enzyme. And so it's very, very important for organisms. The net photosynthesis rate as a function of leaf nitrogen is high as a function of rubisco. Again, all stuff we've emphasized in the past. Where is this stuff coming from? Now, the majority of the nitrogen that we find is atmospheric nitrogen, or N2 gas, totally inaccessible to anything except bacteria. Okay, so besides, like, you know, these eruptions that are releasing this nitrogen gas, and lightning strikes, which are creating nitrogen gas, right? Notice where all the action is happening. It's all happening underground. We have fungus, but mostly it's a bacterial thing. Okay, so what ends up happening is we've got different types of bacteria that have different roles. Some will convert the atmospheric nitrogen into ammonia, some will convert it into nitrites, and some will convert it into nitrates, and they will return it back to the root of the tree, you know, the organism, so that they can uptake it. All right, so this is really what you want to memorize. You see this big lightning strike here. You know, that's where a lot of the nitrogen is coming from, as well as the volcanic eruptions. So when we say nitrogen cycle, I want you to think bacteria, okay? And I want you to also think that, you know, a lot of this is deriving from atmospheric, okay? Where is nitrogen coming from? It's the atmospheric pool, okay? So just like we saw with phosphorus being sedimentary, this is going to be atmospheric, and we're going to focus on the bacteria. Without this bacteria, no plants would be able to access this nitrogen. Okay, so the nitrogen cycle is 4 steps, and we're going to stop at the end of this slide. Okay, the first one is fixation. All right, you have to memorize this section. So when atmospheric nitrogen is fixed, the bacteria are taking N2 gas and converting it into something useful. That's the first step, is they're pulling in that N2 gas. There's a step of ammonification, where amino acids are going to be converted into ammonia. There's going to be a bacterium, a special bacterium, that in the presence of oxygen will convert the ammonia into a nitrite or a nitrate. Okay, so the ammonification, you know, is converting it to your NH4, right? And then you've got your nitrification. When we have oxygen available, we're going to take that ammonia and we're going to turn it into a nitrite or a nitrate, an NO2, NO3. The final step is, let's take those nitrogen compounds that we've created and return it back to the pool of atmospheric nitrogen. And that's denitrification. Okay, so try and commit these things to memory. The bookends are fixation and denitrification. Taking your N2 gas, fixing it into something useful. Denitrification, taking what was useful, converting it back into that atmospheric nitrogen gas. Okay, in between, it depends. If you've got oxygen, that's the nitrification step. That's where we'll convert that ammonia typically into nitrites and nitrates, the ones with the NOs, NO2, NO3. And the ammonification would be that first step where we're going to tack on the H's, right? We're going to convert it into an ammonia form. But again, bacteria underground, the majority of that kind of happening underground, making it useful, a useful form for the plants to uptake, right? But largely an atmospheric source. Okay. The next two slides just talk about the helpers, right? We've got our rhizobia helpers, alder, peas, kudzu, this invasive vine, they all have it, those root nodules that we talked about where the soil bacteria gets taken up, and lichens, right? So anyways, we'll pick up there. We don't have many slides left. We'll pick up there on Friday, but I'm hoping the spouse is going to be able to help us out with the video so we can have a little enjoyable Friday afternoon. So if you want those extra points, hop on that quiz sometime between now and tomorrow, just 4 quick questions, and hopefully you're good for it. My lab, I will see you guys shortly, and we're going to do some data analysis, and I will see you guys on Friday. Bye.
Thank
Bye.
you.
Thank
Yeah.
you.
I just wanted to ask a question about my grades.
Yeah, Mia, can we do that off the Zoom?
Yeah, absolutely.
Okay. I'll send you a link.
Okay. All right. Bye.
Yep, bye.